So in today's lesson, we're going to delve a little deeper into the process of transcription. You'll recall from yesterday's lesson that there are two primary steps to protein synthesis. The first being transcription, where a copy of DNA is made in the form of RNA so that it can leave the nucleus. And once it leaves the nucleus, it's going to go through a process of translation, being translated at the ribosome. So the overall process of transcription involves three basic stages: initiation, elongation, and termination. And I hope to outline each of those for you. So I want us to focus on this diagram that's at the bottom here. So let's just orient ourselves for a minute. So first of all, we can easily recognize, based on the double helix structure here, that we're looking at a molecule of DNA. And so if we look at this molecule of DNA, the goal of transcription is, of course, for us to take our DNA, the section of the gene that needs to be coded for, and to make it into a copy of itself, a single-stranded copy that is in the form of mRNA. And the reason for that is that our DNA is bound to the nucleus of our cell, but our little ribosomes are where the protein synthesis actually happens, is outside in the cytoplasm. So by DNA being converted into mRNA, it provides a way for DNA's code to be able to get to that ribosome. Okay, so now let's take a look at initiation as a process. And I think you'll recognize a lot of similar molecules involved in transcription as you saw in DNA replication. So as in DNA replication, there has to be a way for all of this to get started. And so the process of mRNA transcription happens first when we have an enzyme called RNA polymerase arrive on scene. And, let's think about that name, RNA polymerase, very similar to DNA polymerase 3 in its role. And its role is to make polymers in the form of RNA, right? So, and it's an enzyme as indicated by that ase ending. And so it arrives at a segment just a little bit upstream from the gene that's to be copied. So let's just imagine that here is the segment of our DNA that needs to be copied. Our RNA polymerase is going to arrive just a little bit upstream of that, just a little bit beforehand, in a region called the promoter region. Okay, so think about what a promoter does. It promotes a particular event, right? It facilitates it. And so this promoter region facilitates the transcription of mRNA, kind of helps to get it going. Now, here it is. I've just outlined this here in our note. The promoter region is rich in A and T. So adenine and thymine on our DNA molecule is going to be a segment there that's rich in that. And the reason is it takes less energy to break up A and T as opposed to a bunch of cytosine and guanines, because, of course, adenine and thymine are only bonding by 2 hydrogen bonds as opposed to 3. So because there are a bunch of A's and T's, it is called the Tata box, which is a name that I'm not, particularly fond of, but there it is anyway. Okay, so that Tata box is there. It is recognized by our RNA polymerase, and at that point it binds to the promoter region. And that kind of triggers a few different things. It's going to help to unwind the DNA strand where just a little bit upstream from where the gene needs to be copied, and exposes it so that it's ready to be copied. So that's it. That is initiation. So the next stage, once RNA polymerase binds to that promoter region, we're ready to start making a copy of the DNA in the form of mRNA. So how is it going to do that? Well, it shouldn't be a big surprise to you that complementary mRNA strand is going to be built in the 5' to 3' direction, right? So in a similar way to how we saw with DNA replication. So there's our RNA polymerase just sort of moving along. And as this is happening, it's going to take in our RNA nucleotides But notice that because it is RNA, in DNA replication, we would first need to have a RNA primer to start this whole off, right? Before, almost like a landing dock to get things going. Whereas, in the case of transcription, we don't need that. And the reason we don't need it is because we already have RNA being built here, right? So no primer required. Okay, so we know that only the gene is transcribed, so that ta-ta box that started things off is not part of the gene that's getting transcribed. And as we discussed yesterday, we know that in our RNA, RNA has a uracil as opposed to thymine, right? So that complementary strand, anytime we would happen to see an adenine on the DNA template, the complementary nucleotide would have uracil as opposed to thymine. So that's it. So that's just going to go on and on and on. And eventually it's going to reach a point where there is some sort of a code indicating to stop transcribing the section of DNA. So the RNA strand is going to be, continue to elongate until a section on the DNA, which is referred to as a Terminator sequence. And remember, this is not the same thing as a stop codon, right? The Terminator sequence is on the DNA, whereas a stop codon would be on our RNA. All right, so what happens as a result? Well, that's Terminator sequence. Once identified, lets RNA polymerase know that it's time to stop. Okay, RNA polymerase is released, and so is our new mRNA strand, almost ready to go off and move to the ribosome where it's going to provide the code for a protein. So putting these pieces together, we have, our promoter region called the ta-ta box, which is recognized by RNA polymerase. It's going to attach itself to that promoter region because it's rich in adenine and thymine, so easier to break. RNA polymerase is going to build that new mRNA strand in the 5-3 direction, and it's going to do that until it reaches a termination sequence, which is going to signal to RNA polymerase that the gene is done, we're at the end of the code, and then we're going to release our mRNA. So our mRNA has just been built, it's ready to leave the nucleus and go to the ribosome, right? Wrong. So there are a few processing steps that need to take place before that mRNA is ready to go, because it's been made, but it's just not quite ready to go. So if, I'm going to draw out an mRNA strand. Let's just imagine newly synthesized, and it's going to have a 5' end, and it's going to have a 3' end. The problem is that if we let that mRNA strand go out into the cytoplasm contains all these nasty digestive enzymes that would break it down, right? And you'll remember, so let's say I can draw like a Pac-Man, I guess. Imagine we had these, enzymes eating, taking apart, digesting our mRNA strand. Well, remember, we have all these precious codons that are all along that strand that code for very specific amino acids. So if our digestive enzymes, if these that are present in the cytoplasm go ahead and start to attack the gene that we're trying to code for, that could potentially screw up the entire primary sequence of the protein that might result, right? So that is a big problem. So a few processing things happen. The first is what is referred to as capping. So at the 5' end, what you're going to see is a cap of what's called 7-methylguanosine. Okay, and so I'm just going to draw it in. It's like a cap. And so instead of our Pac-Man digesting our actual mRNA strand, it's going to maybe potentially digest that cap. Now, that cap also plays a role in helping our ribosome to recognize that we want to start translate. There's an mRNA strand, we need to make a particular polypeptide. That cap at the 5' end of 7-methylguanosine will help the ribosome to recognize what the heck is going on. So that's one thing that happens. So a second thing that happens here is that there is also some tailing that goes on. So we've talked about the cap at the top, at the 5' end. At the 3' end, we have a tail that is about 200 adenine nucleotides long. So I haven't really, so just imagine at the end here, let me see if I can make this a little bit, there we go. I would have a bunch of adenine nucleotides all at the end here, and that is referred to as a polyadenine tail. And it has a very similar function to the cap in terms of it just preventing the tail end of that mRNA strand from being digested. Now, the final thing that needs to happen is that we have these things called introns that need to be removed. So what is an intron? Well, an intron is essentially the name that we give to the non-coding regions of our mRNA. So, you may have thought that the entire segment of that mRNA actually is going to eventually end up coding for specific amino acids and therefore specific proteins. However, that is not the case. We have these introns and exons. The exons are the coding part and the introns are the non-coding part. And so we have these exons and these introns. So let's just visualize this for a minute, right? So here is our gene, and so this is our mRNA strand, and we've kind of just, we've coded for, made a copy of the DNA. But within it, we have all these, important parts, which are called exons, but we also have these non-coding regions. These are our introns. So we've already talked about how the cap needs to be there at the 5' end. Our polyadenine tail is going to be there at the 3' end. But also we need to excise all of these introns, and that will give us our mature mRNA that is ready to leave the nucleus. So let's write a little bit of that down. Okay, so we have our introns that need to be removed, and so that happens mostly because that protein needs to fold properly. We know that the positioning of the R groups are amino acids. Depending on where they are along that primary structure, will create different intermolecular interactions. Now, one of my favorite words in biology is this one. It's a spliceosome. I just think that's a really funny name. But what does a spliceosome do? The spliceosome is going to come in and splice out all of our introns. Okay? So mRNA, once transcribed, not yet ready to leave the nucleus, needs to go through some processing, which involves adding a cap to our 5' end, adding a polyadenine tail to our 3' end, and also removing the non-coding introns using spliceosomes. And that is it. That is transcription, at least for this year. You'll notice in some of the videos that I will post to help you visualize this that they may use some additional terminology and reference to particular molecules that are beyond the scope of this course. So don't concern yourself with those. And if you have any questions, be sure to reach out.